class this week we're getting into the chemical basis of life with macromolecules. Okay, the four macromolecules, of course, are proteins, lipids or fats, carbohydrates, and nucleic acids. Right? But central to all of those is this atom called carbon. Okay, carbon is very important. Of course, it's the basis for all those macromolecules. Must have carbon. Okay? The reason carbon is so important. Carbon is stable and really flexible. Stable yet really flexible. Okay, remember, it's relatively small molecule, so it's just got that first energy level filled with 2 electrons, and then the second one has 4 valence electrons. Okay, because it has those 4 valence electrons, okay, it's very, very flexible. Okay? It can form, because of that, form these long chain molecules. We go further than that, and then you can have branches off of it. You have something like a sugar, like this, which is actually a ringed compound. Every one of those little junctures there, angles, is a carbon. Okay, so off each carbon there's a hydrogen off one side and a hydroxyl or OH group off the other. Okay, so that's why C6H12O6. Okay, so form ring structures. Okay, and it can form long chains. Okay, so like here, C3H8, that's propane. Okay, it's a gas like for my barbecue there. Okay, so also brings up hydrocarbons. Hydrocarbons are just like it says, hydrocarbons, just hydrogen and carbon together. Okay, so I'll make the simplest one here, which would be 4 hydrogens, which would be CH4, okay, that's methane, so each hydrogen has 1 electron to share. So they form a covalent bond, they share those electrons. They have about the same electronegativity, so it's a nonpolar bond. Okay, so if you get something, a large hydrocarbon like this, okay, all nonpolar bonds, it's going to be a nonpolar molecule, okay, which we'll get to that a little bit more later on. Right? Good thing about this, there's energy in these bonds, of course. Okay, so methane's a gas, you get energy from that. Of course, propane down there, that's a gas. Hydrocarbon, which you can get energy from. Okay? And the next thing about carbon, which gives a lot of flexibility, is it's got the ability to add functional groups. Okay, so functional groups are groups that you can add on to it. For example, put a little COOH, which is called a carboxyl group, and that's a bit of an acid, that's an organic acid. Okay, you can make it a base by adding a amine group. Okay, so you can add those and change characteristics. You have a methyl group, you have a hydroxyl group, so you can change the molecule pretty easily, okay, by binding with that carbon. All right? Okay, so those are functional groups. Now with all the macromolecules, okay, whether it be a protein, a lipid, carbohydrates, or nucleic acids, they need to work out this premise of a monomer polymer, okay, like a paper clip to a chain of paper clips. You guys probably know what polymers are, but polymers are just repeating units of something. Okay, so for example, with proteins, proteins are proteins made up, or polymers made up of amino acid monomers. So in order to build those up, you need reactions to make those add together. And then sometimes you need to break them down. So to make molecules, whether it's a protein, lipid, carbohydrate, whatever, you use what is called a dehydration reaction. Okay? So you can say dehydration, what are you going to lose? You lose H2O, or water. Right? Okay, so here I've got an example of one. This happens to be an amino acid. You probably know amino acids make up proteins. It's the monomer of protein. Okay? So at the core of that amino acid, you have carbon, of course. We call that the alpha carbon, okay, the central carbon. Off each of those, at carbon, you have a hydrogen group, okay, one bond sites. Off another bond site, you have an amine group, NH2. Okay, that's an amine, that gives it slightly base properties. Off the other end, you have a carboxyl group, or carboxylic acid group, okay, which is a carbon. We double bond to an oxygen to an hydroxyl group. And then that's the same for every amino acid, but off one of the bonding sites you have is called an R group. You probably know there's no atom or element with R, okay? But what an R group is, it can be different things. So it could be an amine group, it could be a methyl group, it could be a hydroxyl group, or it could be a chain of carbons, okay? So it can change, okay? In humans anyway, in animals, it's made up of 20 different amino acids. So that R group could be 20 different things. So it gives each amino acid some slightly different characteristic. Okay, so what I'm going to show here, I have another alpha carbon here. So since it's a amino acid, okay, here I'm going to have that's COOH group here. I'll just put COOH, that's an abbreviation for that. Put the hydrogen here. Okay, and off here I'll put an R group. Okay, and off here, of course, you can predict it's going to be a amine group, and I'll go ahead and write out the structure for the amine group here. Okay, so you have two separate amino acids. Okay, you're building up a protein, you got to put those together. Okay, and that's a dehydration reaction. So if I figure it out, you're going to take the OH from one of the carboxyl groups, and you're going to take an H from one of the amine groups, and you're going to get rid of that. So H2O. Get rid of a water. So water is produced by this reaction. That's why you're removing water. That's why it's called dehydration. And you get a bond. Of course, this is not to scale between those two. And it makes, now you, instead of having two separate amino acids, now you have two. In this case, we call that a dipeptide. They call this bond right here a peptide bond. So you start to build up a protein that way. Okay, and we have a long chain of those. The building up of a molecule. Opposite of that, if you want to split it, of course you probably know you can split proteins and get energy from it. Okay, you can do a hydrolysis. Okay, so again, hydro is water, lyse means to break apart. You can actually just kind of do the opposite. You can take a water molecule, stick it in there, and split that dipeptide back into their amino acid groups. Of course, when you do that, you just do the opposite. So you end up with an OH group here and an H group back here, and you break that bond. Okay. You do release some energy when you do that, and you're back to having two separate amino acids. Class, the first macromolecule I'm going to more in depth are proteins. Okay. Proteins are really, really important because you are what you are because of the proteins that make up your body. Okay. Of course, DNA or nucleic acids hold the code, okay, but that code codes for proteins, which makes organisms what they are. Okay, so really important to understand the concept of proteins. As I mentioned before, made up from 20 different amino acids. Right? So an amino acid is the building block of a protein or a polypeptide. We'll distinguish that in a second. But these 20 different amino acids, of course, differ in that R group, like I mentioned before. But all amino acids have a alpha carbon, an amine group, acid group, okay, an organic acid, and a hydrogen coming off of it. All right, but all these multitude of functions, okay, so I put wow there. A catalyst, okay, you guys remember catalysts from chemistry? Catalysts make reactions go. If I took sugar and put it in my hand, I could wait there all day, nothing would happen to it. I wouldn't burn it in my hand. But in my body, because you have enzymes which lower reaction rates, these protein enzymes, they make reactions go in your body. Okay? There's lots of catalysts in your body. Proteins do that. Defense system, okay, your immune system, you produce antibodies. You guys heard about all the vaccines. See if you produce antibodies. Antibodies have a certain shape, which is going to lock into that virus or bacteria to stop its function, to fight it. Okay, so defense, transport, like blood, hemoglobin is a protein, okay, which is actually a quaternary protein, which we'll talk about in a few minutes. But of course, it takes oxygen throughout your body. Okay. Support, I'm standing here because I have connective tissue, which is made mostly of protein. Bone, you can kind of, you always think of a calcium in bone, but actually there's collagen and protein fibers in bone, in your skin, in your hair. Okay, so support, structures, motion, okay, moving back and forth. Inside your muscle cells, you have actin myosin fibers, which are protein fibers. Okay. Regulation, right? So things like hormones, they trigger certain things. female hormones, male hormones. They turn certain things on and off, so regulation is a bodily system with that. And then storage, of course, you don't always burn. Well, yeah, proteins can be used for energy. Okay, about 4 kilocalories per gram for that. All right. So the thing with proteins we mentioned before. Okay, we break it down into their structures. Okay, and again, structures dictate the function of it. Okay, but they break that down into 4 different levels. Okay. Some proteins only go to the tertiary level, but then other proteins go all the way to quaternary. Okay, so simplicity, what a protein is or a polypeptide is depends on its primary structure. Primary structure is just the sequence of amino acids. Okay, so I have an amino acid here. One of the amino acids is alanine, and they abbreviate acids with just 3 letters, so that's alanine. It may combine with valine, which then may combine with cysteine, which then may combine with methionine, okay, and then on and on and on. Okay, so that sequence of amino acids, that's called the primary sequence. Okay. Secondary is can either be alpha helix or beta pleated sheet. Okay, when you have a alpha helix, it spores this spiral pattern. Okay, and that's between an amine group of one of the amino acids, and then down the chain a ways, carboxylic acid group of another, and they form a hydrogen bond between those, and so you get this characteristic spiral shape. Beta pleated sheet is when you produce the primary sequence and it folds back on itself. Okay, a lot of structural things that's focused in those type of molecules. Okay, then really important is tertiary. Okay, that's the shape of the protein, what shape it becomes. Okay, so you have this long chain of amino acids. You let go of it, it's going to get a characteristic shape. I can just make a shape like this. That might be the shape of a protein. Okay, it's like a key. Okay, that key works because of its certain structure of that key. Okay, same thing with proteins. They work because of their structural shape. Okay, that's what's given by this tertiary shape. Okay, interaction of the R groups. Okay, so these R group interactions, you can have hydrogen bonding. You can actually get some sulfur bonding. You can get some ionic bonds between these different amino acids. Okay, so you can get all these different structural patterns based on the R group interactions. And then quaternary structure. Quaternary is when you have 2 or more polypeptides that come together. Okay, great example of that is hemoglobin, okay, which is used, of course, for transport. Okay. Hemoglobin consists of actually 4 different polypeptides, and they all come together. And actually, a little trivia, they have iron at the center of it. Okay, so you need iron to make a polypeptide, or that protein. Okay, but that's quaternary. Not all proteins have that, but some proteins you have to have 2 or more. 2 or more polypeptides together.